Okay, welcome you all back. So we have already studied what is diffusion, what are the factors affecting diffusion, normal diffusion process. That is also called a simple diffusion. So we have some other diffusion process also that we will see now. That is, next topic is facilitated diffusion. Facilitated diffusion. So see here, by the name only you can understand. ಹೆಸರಲ್ಲೇ ನಿಮಗೆ ಅರ್ಥ ಆಗುತ್ತೆ. It is also a diffusion, right? Facilitated ಇರಬಹುದು, but this is also a diffusion. Diffusion ಅಂದ್ರೆ ನಿಮಗೆ ಗೊತ್ತು. It is movement of molecules from higher concentration to lower concentration, and that is along the concentration gradient. So even for facilitated diffusion also, concentration gradient must be there. So please remember, even facilitated diffusion ಏನಿರಬೇಕು, concentration gradient must be developed already. Must be there. Concentration gradient must be there. Then only facilitated diffusion can happen. But what is facilitated diffusion alva? So see, facilitated diffusion ಬಗ್ಗೆ ಮಾತಾಡಬೇಕು ಅಂದ್ರೆ there are some important things what we need to know. Already ನಾನು ನಿಮ್ಗೆ ಹೇಳಿದ್ದೀನಿ that cell usually has a cell wall, and inner side what it has a membrane called plasma membrane, right? So this membrane is called plasma membrane, and you know very well that plasma membrane, this plasma membrane what we have, so this is called cell wall. This is cell wall, and this one what we see here, this one is cell membrane. So this cell membrane is also called as plasma membrane, right? In plant cell it is called plasma membrane. See here, ಈ ಪ್ಲಾಸ್ಮಾ ಮೆಂಬ್ರೇನ್ ಇದೆಯಲ್ಲ, ದಿಸ್ ಇಸ್, ಇಫ್ ಐ ಝೂಮ್ ಇಟ್ ಅಂಡ್ ಶೋ ಯು, ಇದನ್ನ ನಾವು ಹೈಯರ್ ವರ್ಷನ್ ಅಲ್ಲಿ ನಾವು ನೋಡಿದ್ರೆ ಹೌ ಯು ಕ್ಯಾನ್ ಸಿ ದಿಸ್ ಲೇಯರ್, ದಿಸ್ ಪ್ಲಾಸ್ಮಾ ಮೆಂಬ್ರೇನ್ ಹೆಂಗ್ ಕಾಣಿಸುತ್ತೆ ಅಂದ್ರೆ ದಿಸ್ ಇಸ್ ಹೌ ಇಟ್ ಇಸ್. You might have observed, ಅಥವಾ you might have studied about this model. We call it as Singer and Nicholson's what is this, plasma membrane model anta kariತೀವಿ, fluid mosaic model anta kariತೀವಿ, right? So plasma membrane, if you see, actually it is like this. You know, it is a lipid bilayer. Andre this is also a lipid layer, this is also a lipid layer. Two lipid layer together will form a one plasma membrane. So it is lipid bilayer, right? So two layers of lipids are there. So ಏನಾಗಿದೆ ಅಂತಂದ್ರೆ, this when we study the cell cycle, cell the unit of life ಅಲ್ಲಿ, in that chapter we will study in detail. But try to understand one thing. Plasma membrane is made up of lipid bilayer. It is completely made up of lipid things. Okay, we have ಈ ಕಡೆ side ಏನಿರುತ್ತಲ್ಲ, this surface is water loving surface, this head, but tails are hydrophobic. Hydrophilic, hydrophobic, these are the two terms you need to understand. Andre because membrane is made up of lipid, then obviously it will have affinity towards lipid molecules. So ಇದನ್ನ ಹೆಂಗ್ ಅರ್ಥ ಮಾಡ್ಕೊಬೇಕು ಅಂದ್ರೆ, whenever you put water into water, water will dissolve water. It will easily mix off, right? But if you put oil into water, mix agutta? No, it will not mix. Instead it will start, what you can say, floating on the surface. Same thing, if you put water into oil, that also will not take it, because water hates oil and oil hates water. Those both will not easily get mixed off. So in the same way, illenagutte antandre, but if you put oil into oil, then obviously it will diffuse easily into that oil particles, and water into water, that will also easily diffuse properly. Likewise, because plasma membrane is made up of lipid, okay, water cannot easily enter this lipid, because it will start hating that water. It doesn't want that water to enter inside. In the same way, all the substances which are water loving, that also is not allowed inside. So all the water loving things are called hydrophilic. So we call the terms, these are the terms. So hydrophilic andre eno, hydro means water, philic means love. So water, philic andre loving. So what is hydrophilic substances? Water loving substances. Now those water loving substances, they love water, but this lipid layer will hate that water loving substances. Alva, lipid is a bilayer of, this membrane is lipid bilayer. It is made up of lipid. It will easily take all the lipid molecules inside, but it will not allow water loving substances to pass through them easily. even though there is a concentration gradient, it cannot easily pass through this membrane. As I've already told you that membrane plays an important role as it gives permission. As a permission kodlilla andre adu enter agalla. Likewise. So what needs to be done? What can be done? You know that plant is completely made up of water. Now water loving substances is jasti irutte plant alli. Then if this membrane is not allowing those molecules, then what will be the condition, right? Somehow we need to facilitate the movement of those molecules, movement of those hydrophilic molecules. So when you allow the movement of hydrophilic substances through this lipid layer, lipid bilayer, then that is also called diffusion. Andre movement of hydrophilic substances from higher concentration, let us consider this is higher concentration, they all are hydrophilic, okay? If they have to move into this, then this is called diffusion only. Higher concentration to lower concentration. But because it is hydrophilic, it is not easy to pass through the membrane. What we need to do is, we need to facilitate it. ಅದಕ್ಕೊಂದು facility ಕೊಡಬೇಕು ನಾವು ಹೆಂಗಾದ್ರೂ ಮಾಡಿ ಅದನ್ನ ಅಲ್ಲಿ pass ಮಾಡಿಸಬೇಕು. For that reason, what actually can be done is, if you study the structure of lipid bilayer, you might have seen or you might have witnessed, there are some channels which are present here. There are some channels which are present here like this. These are called channels, proteins. These are nothing but proteins like this. These are called completely lipid layer only. If you see so many areas, you will have this proteins like this, and these proteins are called transmembrane proteins, or normally transport membrane proteins, transmembrane proteins anta karitivi. So what this transmembrane proteins will do, these will help in the movement of this molecule through this layer like this. So because this transmembrane protein, I will write what it is called. It is called transmembrane protein, right? So now this transmembrane protein, because it is facilitating the movement of these molecules to pass through it, so this diffusion is called facilitated diffusion. So I hope I made it clear what is facilitated diffusion. One facility ಕೊಟ್ಟು ಅದನ್ನ ನಾವು ಕಳಿಸ್ತಾ ಇದೀವಲ್ಲ one side ಇಂದ ಮತ್ತೊಂದು side ಗೆ, it is diffusion only, movement of molecules from higher to lower, but with the help of transport membrane protein. So we call it as facilitated diffusion. And this is must, because lipid layer is lipid loving. It will hate water loving substances. So if water loving substances should enter inside, then there should be a special facility to take it through that. So this is called facilitated diffusion. I hope what is facilitated diffusion, I hope it is clear now. So what you can remember in this facilitated diffusion andre, now rate of this facilitated diffusion, again idike ಏನೇನು important I told you, concentration gradient must be there already, andre matra it can move from higher to lower. So because it is also a diffusion only, then what is required here? It requires a transport membrane proteins. So these proteins are important for the movement of these molecules, right? So transport membrane proteins are required. Second thing, which molecules can diffuse faster? Always the size, it matters. Size of the molecules. Slower the molecule size, lesser the size ಕಮ್ಮಿ ಇದ್ರೆ movement will be fast. Size ದೊಡ್ಡದಿದ್ರೆ molecules movement will be slow. So size should be smaller will move fast, larger will move very slow. You people know it very well, size of the molecules. Next is solubility. Solubility of the molecules. Andre, as I told you now, just molecules, if they are water loving, then it is very difficult to pass through it lipidally. If it is lipid loving, then there is no need of this proteins at all. Aram se move agutte. If it is lipid loving. Water loving idre facilitation should be done. So we call it as facilitated diffusion, right? Now, characteristics of facilitated diffusion is again the same like diffusion process only. Same characters you can write here also. But the thing what you need to remember, there are three important terms here which is not there in the simple diffusion. Those three important terms, please remember, we call it as 3S anta karitivi. We call it as 3S. First thing is, they are selective. Second thing is they can achieve saturation. Third is sensitive. Selective, saturation, sensitive. So these are the three important terms actually which we need to study regarding facilitated diffusion. So we will try to understand everything. So first thing is selective. As I told you, there is a special membrane proteins through which the molecules move. You can say that it is selective anta helbodu. That means it knows ee proteins ge goತ್ತಿrutte what should be taken inside, what should not be taken inside anta. So that is called selective anta ಹೇಳ್ತೀವಿ. Right? So it is very much specific antanu helbodu. You can also call it as specific. It knows what to take inside, what not. So which molecules are important to the plant? Only those molecules are taken inside. Andre, even along with this, for example, let me show you some other molecules. Like, along with these molecules, even these molecules are there. Anta tilkodi. Here, these molecules are less. Along with this, even this should go actually, alva. But if plant does not require that, then these molecules are not taken inside. Proteins are specific for specific molecules. We have particular proteins for particular molecules also. So we call it as selective or specific anta karitivi. Okay. Then next is saturation. Saturation andre enu? What do you understand by the term saturation? Means complete level is done. Completely it is done anta helbodu. Like, for example, if I take a glass of water and if I start adding sugar into it or salt into it, see, initially sugar ಏನಾಗುತ್ತೆ ಕರಗ್ತಾ ಹೋಗುತ್ತೆ, right? So ಹಾಕ್ದಂಗೆ sugar ಹಾಕ್ದಂಗೆ ಅದು ಕರಗ್ತಾ ಹೋಗುತ್ತೆ. ಎಲ್ಲಿವರೆಗೂ ಕರಗುತ್ತೆ? ಎಷ್ಟು ವಾಟರ್ ಮಾಲಿಕ್ಯೂಲ್ಸ್ ಇದೆಯೋ ಅಷ್ಟು ಶುಗರ್ ಮಾಲಿಕ್ಯೂಲ್ಸ್ ನ ಹಾಕೋವರೆಗೂ ಅದು ಕರಗ್ತಾ ಇರುತ್ತೆ. ಬಟ್ ಲೇಟರ್ ತುಂಬಾನೇ ನಾನು ಶುಗರ್ ಹಾಕ್ತಾ ಹೋದ್ರೆ ಫೈನಲಿ ಇಟ್ ವಿಲ್ ಕಮ್ ಟು ಒನ್ ಸ್ಟೇಜ್ ವೇರ್ ಶುಗರ್ ಡಿಸಾಲ್ವ್ ಆಗಲ್ಲ. It will stop. Even though if you are adding, it will not get dissolved completely. ಯಾಕೆ ಅಂತಂದ್ರೆ ಸ್ಯಾಚುರೇಷನ್ ಲೆವೆಲ್ ರೀಚ್ ಆಗ್ಬಿಟ್ಟಿದೆ, ರೈಟ್? ಸೊ ಹೊಟ್ಟೆ ಹಸು ಇರೋವರೆಗೂ ನಾವು ತಿಂತೀವಿ. ಒನ್ಸ್ ಮೈ ಸ್ಟಮಕ್ ಇಸ್ ಫುಲ್, ದೆನ್ ಐ ಕೆನಾಟ್ ಈಟ್ ಎನಿ ಮೋರ್, ರೈಟ್? ಸೊ ದಟ್ ಇಸ್ ಕಾಲ್ಡ್ ಸ್ಯಾಚುರೇಷನ್ ಲೆವೆಲ್. ಆ ಲೆವೆಲ್ ಮೀಟ್ ಆಗಿದೆ ಅಂದ್ರೆ ದೆನ್ ಆಫ್ಟರ್ ದಟ್ ವಿ ಕೆನಾಟ್ ಈಟ್ ಎನಿಥಿಂಗ್. That is called saturation level. But andre you can relate it with that example. But then what is saturation in this case? Saturation andre, now when you see this plasma membrane like this is a plasma membrane for example. Then we have, for example, we have protein channels over here. We call this transport membrane proteins as channels also. We can call them as channels also. So, how many are there here? 1, 2, 3, 4, 5, 6 channels are there, for example, or 6 proteins are there. Now, initially I have 6 different molecules or 6 same molecules. Or I can say I have some more than some 13 or some molecules are there. Initially diffusion will start because if permission is there with this transport membrane protein, then movement can be done, no? Initially what will happen? Movement of these molecules will start. One will enter inside, 2 will enter inside. So each time only, initial starting time, if you see, diffusion takes place very fast. Because this molecule has opportunity with this. This can go from here. This can go from here. This also has opportunity from here. So initially ಏನಾಗುತ್ತೆ ಅಂದ್ರೆ when we start facilitated diffusion, molecules are more. How many we have? 6 channels we have. So initially 1, 2, 3 ಅಂತ ಮೂವ್ ಆಗ್ತಾ ಇರುತ್ತಲ್ಲ, initially it is faster. But when all the transport channels are open, now this also is entering from here. Now at a time when the 6 molecules are entering inside, ಎಲ್ಲ channels open ಆಗಿದೆ. Now the rate will decrease because all the channels have opened. So faster ಆಗಲ್ಲ because ಎಲ್ಲ ಕೆಲಸ ಮಾಡ್ತಾ ಇದೆ. There is no free transport protein here. So once, even though concentration ಜಾಸ್ತಿ ಇದೆ ಹೊರಗಡೆ, but now we want to take more more molecules inside, but it is not possible. Why? Because already all are open, all are doing their function. So it will take time until all the molecules, all the 6 molecules will enter inside, then only it will give time to the next molecules, right? ಸೊ ಇನಿಷಿಯಲಿ ನೋಡಿದಾಗ 1, 2 ಅಂತ ಎಂಟರ್ ಆಗಬೇಕಾದ್ರೆ ಸ್ಪೀಡ್ ಆಗಿ ಹೋಗ್ತಾ ಇರುತ್ತೆ, ಬಿಕಾಸ್ ಇದು ಓಪನ್ ಇರುತ್ತೆ, ಇದು ಓಪನ್ ಇರಲ್ಲ, ಅವಾಗ ಓಪನ್ ಮಾಡಿ ಒಳಗೆ ಹೋಗುತ್ತೆ. ಸೊ ಇಟ್ ವಿಲ್ ಟೇಕ್ ಟೈಮ್ ಇನಿಷಿಯಲಿ, ಬೇಗ ಆಗುತ್ತೆ. ಬಟ್ ಲೇಟರ್ ಆನ್ ಏನಾಗುತ್ತೆ, ವೆನ್ ಆಲ್ ವಿಲ್ ಓಪನ್, ವೆನ್ ಆಲ್ ದ ಚಾನಲ್ಸ್ ಓಪನ್, ದೆನ್ ಸ್ಯಾಚುರೇಷನ್ ಲೆವೆಲ್ ಇಸ್ ಮೀಟ್, ಅಂದ್ರೆ ಸ್ಯಾಚುರೇಷನ್ ಲೆವೆಲ್ ಅದು ಬಂದ್ಬಿಡುತ್ತೆ. ಸೊ ವಿ ಕಾಲ್ ಇಟ್ ಆಸ್ ಇಟ್ facilitated diffusion easily gets saturated. When it gets saturated, when all the transport membrane proteins open, or you can say when all channels are open. ಯಾವಾಗ ಅಥವಾ ಫಂಕ್ಷನಿಂಗ್ ಅಂತ ಹೇಳ್ಬೋದು. When all channels are functioning, then it attains saturation level. This is about saturation. Now what is the meaning of sensitive? So you know what is sensitive, at least you can relate it. Sensitive means, see up, these channels are nothing but proteins. They are nothing but proteins. Now there can be some chemical substances when we provide it to a plant by any means, that chemical substance can destroy, or destruction can be done of this protein. You know that proteins easily get denatured at high temperature, high high intensity of what you can say, heat or something. So when it gets denatured, then it will not function properly. So sensitive yak ಹೇಳ್ತೀವಿ ಅಂದ್ರೆ, there are some chemicals, chemicals, we call them as inhibitors ಅಂತ ಕರಿತೀವಿ. We call them inhibitors. So these chemicals, usually these inhibitors can destroy the structure of this protein. And once the structure of protein is destroyed, then obviously movement of things and all will not happen, because the structure only is not proper. So ಅದಕ್ಕೆ ನಾವು ಏನಂತ ಕರಿತೀವಿ, ಸೆನ್ಸಿಟಿವ್ ಅಂತ ಕರಿತೀವಿ. ಯೂಶಲಿ ಟ್ರಾನ್ಸ್ಪೋರ್ಟ್ ಆಗುತ್ತೆ ಟ್ರಾನ್ಸ್ಪೋರ್ಟ್ ಮೆಂಬ್ರೇನ್ ಪ್ರೋಟೀನ್ ಇಂದ, ಬಟ್ ಇನ್ಹಿಬಿಟರ್ಸ್ ಏನಾದರೂ ಹಾಕಿದ್ರೆ ದೇರ್ ಕ್ಯಾನ್ ಬಿ ಸಮ್ ಪ್ರಾಬ್ಲಮ್ ವಿತ್ ಸ್ಟ್ರಕ್ಚರ್, ಅಂಡ್ ಸ್ಟ್ರಕ್ಚರ್ ಪ್ರಾಪರ್ ಆಗಿ ವರ್ಕ್ ಮಾಡಿಲ್ಲ ಅಂತಾರೆ, ದೆನ್ ಇಟ್ ಕೆನಾಟ್ ವರ್ಕ್ ಪ್ರಾಪರ್ಲಿ. ಸೊ ವಿ ಕ್ಯಾನ್ ಸೇ ದಟ್ it is sensitive for inhibitors. So these are the important 3 terminologies which we need to remember. Idu diffusion alli baralla, these 3 things we are discussing this in facilitated diffusion. So 3 important terms of facilitated diffusion. Rest all is same like diffusion. I hope facilitated diffusion is clear. Thank you.